Well, welcome everybody. It's unfortunate again that Steve wasn't here to kick off this event. I kind of relied on some of his practical applications of cover cropping in his research program out of ARS in Maryland. But as Rex mentioned, my name's Jennifer Kucera, and I'm the West Regional Soil Health Team Leader. I'm based out of Portland, Oregon, and I'm part of the new Soil Health Division. We're a team of about 20 that are distributed across the US, and so we represent NRCS, but we collaborate a lot with ARS, farmers, ranchers, land-grant institutions, and scientists and extension agents. So with that, my talk today is going to focus primarily on the soil biology and give some examples about how cover cropping can influence and how management, we can manipulate that microbiome. And I was really excited to hear the plenary speaker today mentioned the directive to really emphasize and ramp up the soil microbiome research, because I think that they really hold a lot of the insights and the keys to sustainable management. So a healthy soil is really a community of soil life. And the way I kind of define a healthy soil is as a living system that supports a diverse, active biological community. And this biological community, right, is the most diverse ecosystem on Earth in the soils, and it's made up of billions and trillions of bacteria and miles of fungal hyphae and all of the meso and microfauna that interact collectively with plants, the soil, and the animals that exist. And they work collectively to aid crop production. And by the end of this talk, hopefully you'll have a better appreciation for the importance of soil biology in providing the essential functions that agriculture is really dependent upon. And these functions that support plant productivity is really central to the decomposition of those residues and forming soil organic matter. So soil organic matter, right, is one of the key soil quality, soil health indicators that we use. It's responsible for driving a bunch of physical, chemical, and biological properties, water holding capacity, nutrient holding capacity, the resistance against erosion and aggregation, et cetera. But it's really the actions of the biology that works as the residues are broken down and converted into organic matter that sets this off. It's also, the microbiome has been classified as the eye of the needle through which all nutrients pass through. So by enhancing nutrient cycling through enhancing organic matter and stimulating the microorganisms, we can actually have a pool of long-term, more sustainable nutrient cycling. Soil structure and aggregation is often thought about as a physical property of soils, but really it's driven and created through the actions of those bacteria and fungi that work together as they break down those residues, form organic matter, literally bridge soil particles together through hyphal networks. And bacteria also are very important in this process as they release these sticky substances called extracellular polysaccharides. And they work similar to glues in the soil. So it's not just fungi, but also bacteria that play a pivotal role in improving soil structure and aggregating those particles together. And that's important, again, for resisting erosion, as well as facilitating improved nutrient or water infiltration, retention, and nutrient holding capacity. The soil life is also, of course, important for protecting plants against diseases and pests. They're also the causal agents of some of those, so trying to balance the good guys versus the bad guys in the soil is a key attribute of the functions. Protecting plants from stress. So it was mentioned earlier with extreme weather events, drought, the incidence of drought increasing, high extreme temperatures, intense rainfall events. The microbiome can actually facilitate and communicate with the plants that can help build that resistance and resilience into the system to help those plants deal with the stresses from the environment. And lastly, of course, they're involved in detoxifying pollutants. So I would argue that all of the functions that we rely on in the soil to support plant productivity are really directed at the soil life. The biology drives the physical and chemical properties that are important in supporting all of these key functions. That's not to say that mineralogy, soil type, texture does not influence and play an important role. It absolutely does, and it creates these thresholds from which the biology can work under. So in soils, again, soils are the most biologically diverse ecosystem, and microorganisms are found everywhere in the most extreme environments, from the Atacama Desert to deep thermal sea vents to Antarctica. But for most agricultural soils, there's an optimal range of conditions where things are just right. And so I like to kind of equate it to the Goldilocks story, where pH, it's not too acidic, not too alkaline. Temperature, it's not too hot, not too cold, it's not too wet, not too dry. Everything is kind of just right. And so for most agricultural systems, biology is optimized at that near neutral pH. That's similar to plants in that most nutrients are most available in that pH of near neutrality. Moderate temperatures, moist conditions, aerated conditions, and in agricultural soils, oftentimes carbon or food, that energy source, is some of the most limiting resource that microorganisms have to adjust for. And because of this more narrow window from which most organisms, the biomass and their activities are kind of confined to, most soil organisms are at rest. So bacteria, about 90% of them are inactive at any one moment. And that's because they're not in direct contact with an organic carbon source, for example. Moisture conditions aren't quite right, pH, et cetera. But there are ways to sort of waken this soil microbiome. One of them, and movement, is through bacteria on the fungal highway. And I don't know if this video will work. Well, all it shows, so this is a fungal hyphae here, and if the video was working, there's little bacteria that are kind of moving at rapid speed along the hyphal networks. And so as the hyphae extend into the soil, they're releasing different enzymes, they're breaking down organic materials, and they help. I wouldn't worry about it, it's okay. And they're helping to translocate and move bacteria through the soil system. Also, to awaken the sleeping beauties, the bacteria in the soil. The Prince Charmings can also be these higher organisms, mentioned fungi, but earthworms have been known to farm microorganisms, and so they can awaken as they pass through the guts of earthworms. They can be translocated from one area in the soil to another, and also help with nutrient cycling and detoxifying different chemicals. Nematodes, other fauna, are really the Prince Charmings, right? They can help awaken and motivate the bacteria to come to life, to then instill all of those different functions that I mentioned at the beginning. So what about management, though? How can management influence and help support all of these higher organisms to facilitate the microbes, the bacteria and fungi, the protozoa and the nematodes that are really driving a lot of nutrient cycling and population regulation? So most organisms are in an inactive state, but there are some biological hotspots. This is where life and action are kind of really stimulated in the soil. And there's 5 key biological hotspots that we like to talk about. The first and kind of foremost is the rhizosphere, right? This is the zone right around the roots. And the abundance and activity can be anywhere from 10 to 100-fold greater at that zone, just a couple of millimeters away from the root surface, than in the bulk soil. And again, different roots have different architectures. They're exuding different chemicals that help stimulate different populations in the soil. So that's one of the hot spots. This is like the nightlife. This is where all the cool kids hang out you want to help support the rhizosphere in your soil. The litter layer then, as these plants die and are decomposed, that litter layer then not only protects the soil from erosion and temperature and moisture extremes, but it also helps facilitate and it acts as a hot spot, right? So earthworms come up, they take residues, they bring them back down, translocate them deeper into the soil. Along with that, nutrients and mineralization of those nutrients can be facilitated. And so the litter layer acts as a biological hotspot, more for some of the macro and mesofauna, centipedes, beetles, earthworms, etc. As these the rhizosphere and the litter layer help stimulate the breakdown of those organic residues, they help to form those aggregates that are so important, again, for infiltrating water, water storage, as well as resisting erosion. And so the aggregate surfaces also act as a hot spot, where the aggregate is really the microbial home or habitat that we want to support. But they also move through the pore spaces. So the half of the soil, right, is made up of just space and soil that acts as sort of the lungs and circulatory system of the soil. So we want space. Space is needed. And then joining those spaces are the aggregates that are supported. And then lastly, these earthworms and root channels, biopores, these larger pores, also are zones of biological hotspots. And in this case, this root is growing in a former earthworm channel that you can see has a darkened material that the deposits from the earthworm are very high in nutrients. The pH is usually neutral. There's also a lot of bacteria that have been transported through there. And so the root not only is following a path of least resistance, but it's also taking advantage of all those great nutrients and biological activity that is formed as that earthworm channel becomes unoccupied. So these are the hot spots that we want to try to maximize. So if we can maximize these biological hot spots, we reduce the voids in the soil, these places of inactivity, and we can stimulate all of those biological drivers of organic matter formation, nutrient cycling, detoxification, plant protection, resiliency, resistance to environmental and abiotic stressors. So we want all of these 5 different hotspots to be optimized through our management practices. So building on this, and I try to equate soil health with human health, and there's some real direct linkages, right, between there's a lot of momentum with the gut microbiome efforts and what you are, what you eat kind of thing, and how microbes that live on, in, and around us influence our health in both beneficial and negative ways, depending on conditions. Similarly, in the soil, microbes that live on, in, and around plants also influence the health of the plant and plant productivity. And so soils are the foundation for nutrients, right? The nutrients that are taken up come from the soil, come from the release of these elements through microbial processing. Then we consume them. So how well we take care of the land and how these nutrients are supplied to our plants is critical for our own nutrition. And then what are the linkages between how we manage our livestock, how we manage our plants, how we manage everything that then translates into our own gut microbiome and our own human health. And so there's a lot of momentum and research going on into identifying those linkages, even within the animal itself and how the animal microbiome helps facilitate and induce different resistance to pathogens and diseases through various strategies of animal health. So how the whole system, and so SARE is so wonderful in really propelling this concept of systems integrated research, right? All of the pieces kind of fit together into a puzzle, and you can't really take one piece in isolation and expect to understand the entire system and how to optimize it. So I really commend SARE in kind of pushing that envelope of integrated systems level research. When I started at Texas Tech, back when I was a researcher, I was lucky enough to get, one of my first grants was a SARE grant, and that we worked on integrated crop livestock systems and how you didn't have to go all in to convert to a perennial system, but if you took a piece of your field and converted it to a perennial or pasture system for livestock integration, it increased water use efficiency, nutrient use efficiency, it built organic matter, and again, my role then was to explore how the microbes facilitated those linkages. So again, I really can't emphasize research from a systems level, putting all the pieces together, build teams that build from economics and social sciences, as well as the biology, chemistry, physics, physiology, plant breeding, genetics. Like, you need the whole team to really understand and move the whole industry forward. So that integration is so important, and you all know that because you're here, and this is a SARE conference, but I just wanted to emphasize the importance of that again. So how do you go about developing these soil health management systems to optimize biological functions? And there's different iterations of this. This is a new book chapter that I was involved with recently that's in press. But success really stems from your knowledge base and truly understanding the history, that management history of the farm or ranch, the forest, and then making sure that you're site-specific, regionally specific, and goal or problem-oriented in your approach. So you just don't throw the book at it and hope that things work out, but you really identify what problem is occurring on the farm, what is your goal, and then you design around that system to optimize it. To do that, you need to do some kind of initial assessment. There are a variety of different ways. Again, that history and really talking with the farmer and rancher and understanding where they're coming from, what experiences they have had, where their successes are, where some of their challenges are is so important. Field and laboratory assessments are wide, and there are a variety of different indicators that we use to do this type of assessment. Probably your number one tool is your shovel, right? So get out, walk the field, dig in the soil, look at it, and identify some of those trouble areas. You can learn a lot just from digging. But that can only take us so far, and sometimes we need some laboratory assessments to build on that, and I'll focus on the microbiome here in a little bit. And then you have to kind of keep your expectations in check. What is your threshold? What are the environmental constraints, your soil type, your climate? What are the resources that you have on hand? And then develop some reasonable expectations and a reasonable time frame for which to achieve those expectations. And so you really have to kind of know your baseline, and that's part of what we're working on with NRCS and ARS to identify a soil health baseline across the country. And then you can kind of know where you're going. And then once you do all of these steps sort of in sequence, you can then identify management goals, both in the short and the long term. And also, I kind of recommend just being patient with yourselves and explore. You might just take a small piece of land and play around with it before you scale up larger. And again, kind of equating to human health and soil health, I think of it as soil health and management practices. You can take it as a fad approach and hit the newest, hottest diet, or you can kind of take a holistic approach and change your entire lifestyle, right? And you wouldn't start, changing your exercise program by going out and running a marathon tomorrow. You might start walking and then running a mile, running a couple of miles. You might not, just all of a sudden stop eating whatever whatever is your temptation, but you might start cutting back. And so it's a whole perceptive of trying to take small steps, be patient, and then assess and adapt and then change, based on how the feedbacks are on your farm, similarly, that you would with your body. When you don't do that and you take shortcuts, what happens if you go out and you run 10 miles after not running for 2 years? You're gonna be really sore, you're gonna be in pain, you probably spend a lot of money, and if you're not, you're gonna go spend some money at the doctor's office for, trying to fix your knee or your ankle or whatever you just messed up by pushing yourself too far, too fast. So same with soil health, it's a journey. It's not something that happens overnight, and you need to take it in smaller chunks to be successful. And then again, reassess and adapt, change based on the feedbacks that you acquire through your assessments. So NRCS it has 4 soil health principles that I really believe have universal applications. Soil health is the same no matter where you are in the world. And you're trying to optimize these efficiencies, you're trying to maximize productivity while maximizing and protecting the environment from which you're working in. And so the 4 soil health principles, you've probably seen these a million times, is to minimize disturbance, maximize soil cover, maximize biodiversity, and maximize the continuous living roots. And really, it kind of boils down to 2 principles in my book, and that's protect and feed. And once we think about soils as a living, breathing ecosystem in and of itself, of all those biological organisms that are doing everything that we rely so heavily on, we then begin to change our view in how we approach our management strategies. So we protect the soil aggregate, that microbial habitat or home, and we protect the organic matter that is so important, not only for food and fuel for them, but for all of those other properties that I mentioned earlier. So this side, these 2 principles kind of get at the protection of soil aggregates and organic matter. And the left-hand side, maximizing biodiversity especially, and maximizing the presence of a living root in your system, is where we can begin to feed, fuel, and build soils. And biodiversity can happen in a variety of ways. Crop rotations, cover cropping mixes are one way, but also integration of animals into our ecosystem is another way. And it can happen at different scales, just having pollinator strips and working in that regards if you're in a pivot circle, for example, the corners, how you manage the corners can provide wildlife and pollinator habitat. Or it can, integrate cattle into your system as another example. So again, protecting the habitat, we're maintaining those organic matter and aggregates, we're reducing erosion and runoff risks, so we're influencing both water and air quality at the same time when we do this. We're buffering temperatures, we're reducing evaporation, we're capturing every drop of water as much as possible and maintaining it in that soil profile for storage later for that plant to take up when things start to dry out and become more stressful. The left-hand side then is feeding and fueling that soil biology with diverse carbon resources. So just like you might get bored and not be so healthy if you ate the same thing day in and day out, we can stimulate different microbial populations by diversifying those sources. And when we do that, we tend to break disease cycles, we're stimulating that microbial diversity, we're increasing organic matter and nutrient cycling, we're enhancing plant growth, and we're increasing predator and pollinator populations. And there are a number of different practices then that can get us there. This is just a very small list to practices that are minimizing disturbance, and this is primarily centered around tillage, but let's not forget about our pest management and fertility management practices, as different fertility regimes can influence that microbiome as well in both negative and positive ways. And then the rest you can see are more from the crop management perspective, and cover crops kind of tend to fall into 3 out of the 4 categories right out of the gate. So cover cropping is one of the nice practices that you can consider to get you started once you've identified ways to minimize that disturbance level. And so there's different entry points for soil health management. There's the crop and soil level where plant choice can really influence. And I'd really like to kind of make a plug or a push for breeding efforts to really identify the variety trials and the crop selection process in systems that are under soil health management strategies. So varieties might be completely different if you grow those varieties in a cover crop system than as opposed to a bare field that are more commonly done in variety trials. And of course, those soil health management strategies that I just outlined are entry points at the crop and soil level. And then there's the plant genetic and microbiome level, where we can look at different ways for disease suppression or inhibiting nitrification or denitrification to keep that nitrogen pool in the place. Inoculation has some really great potentials. There's been great advances with nitrogen fixing bacteria and AMF. There's some new biologicals on the market with varying successes, but something to keep on the radar. But more so than not, what management strategies can we develop that foster disease suppressive, nutrient enriching populations? So then the second half of the talk, I just want to give you some examples of some research that's happening more from that cover cropping perspective, and how management can influence and alter the microbiome and their associated functions. So this is some work out of ARS in the Dakotas, Mike Lehman's lab, and they grew different species of cover crops, and they grew these in pots, and they used the same soil from a native prairie nearby. And the different colors on this chart are different types of fungal functional groups. So some are animal pathogens, some are saprotrophs, those are organisms that break down dead stuff. Some are plant pathogens, and some, the yellow is arbuscular mycorrhizal. So different types of cover crops stimulate and support different fungal functional groups. So for example, clover supported the greatest concentrations of AMF, whereas oats had the greatest concentrations of saprotrophs, and then wheat kind of had the greatest overall diversity. So how can crop selection... Arbuscular mycorrhizal fungi, so these are a symbiotic relationship between the plant root and fungi, which then kind of extend out into the soil environment, increasing the surface area, and they can release special enzymes that are really good at extracting phosphorus from the soil, as well as water. So in those cases, if I was a plant root and then you all are fungal hyphae and we all joined hands, if I'm just a plant root, I get this area, and if we all join hands, then we have the whole entire room to extract water and nutrients from. So AMF is important there. Yeah, okay. So then variety selection. So the point of this, all of these are different bars or different types of organisms. In this case, it's bacteria. So these are 3 different types of corn varieties that support different bacterial communities, and sugar beet really kind of changing everything up. Fertilization can change the food sources. So this study, these different bars are different root exudates. So different levels of nitrogen change the different composition and quantity of the exudates that are coming out of the plant roots, and that stimulated an entirely different population in these different treatments. This is another with different fertilizers, unfertilized, different bacteria, organic fertilizers, inorganic, and then different crop rotations influencing bacterial populations. Amendments, so this is different bars of bacterial populations and native, and then this was in a fumigation trial, so fumigation decreases the diversity that's intended, to try to hit the pathogens, but you can also get there with different composting communities. And so what happened in this study, then they introduced a pathogen, and they found that the fumigated soils reduced the diversity and such in a way that they weren't able to kind of fight off the pathogen that was reintroduced. But the vermicompost and the different levels of composting actually had this high diversity that had the population that when that pathogen was introduced, they actually was able to fight it off. You can also influence the microbiome by different vegetative stages. So this is seeding, vegetative, and bolting. And I don't remember the crop they use, but the point is that different types of organisms have different concentrations depending on maybe termination timing or plant developmental stage. And lastly, this is just some new research out of Dan Manter's lab in Colorado, where they grew different cover crop species, and this is disease severity. So they found sorghum Sudan had the lowest disease severity, they had the highest microbial biomass, and then they started looking at genes that were responsible for inducing this disease suppression, and they found this gene that is important in siderophore production. It's important for nutrient acquisition, as well as changing the chemistry of the plants that then lower the disease incidence. And so getting at that function, not just who's in the soil, but what are they doing and how are they contributing to reducing plant stress is really the direction that needs to happen. So cracking the microbial molecular code for soil health. Soil health assessments to date kind of ignore the microbial and specifically the molecular side of things, but I argue that the molecular information can provide information on all of the key functions that we talk about: organic matter cycling and decomposition, aggregation, nutrient cycling, disease suppression, and stress resiliency. And I won't read these, but suffice it to say that there are genes and different taxa that are really driving a lot of these functions. And if we really want to understand how soil health it can be enhanced under different conditions, we need to understand what the biology is doing. And to decouple those, I think, would be a real oversight. So, in summary, I love soil, but I really think you should say I love soil microbes on those stickers. Soil health is driven, right, by the actions of soil microbes and biota. So sampling and our methodology, our assessment tools, need to focus on the living soil community and that living soil component. We need appropriate databases to evaluate that. Wouldn't it be cool if there was a database where all the different cover cropping trials and the microbiomes and the different communities that popped out and their functions could all be compiled in one area so that we could then tap into that database and design specific kind of designer cover crop mixes to address different functions beyond what we know from the plant perspective. We need a national living soil archive, and ARS is working hard to try to get that. There was a recent opinion paper in PNAS of why we need a national soil archive. And then another kind of idea is an online public forum or discussion group. So this database with lots of cool scientific information could be very helpful, but what about farmer to farmer? What is your experience in your cover crop mixes, your termination time, your seeding rates, on and on? Kind of like a recipe kind of blog, forum where different recipes and then you tweak it and you comment on. Wouldn't that be cool if that could happen to really push the envelope forward? And with that, I'm out of time. But really, we need to change our perspective and get our heads under the ground. That's always been kind of thought of as a negative, but man, we need our heads underground. We need to listen to the soil. And I love this quote by Henry Ford: whether you think you can or you can't, you're right. Right? So have a positive attitude and be patient and understand, your expectations and your thresholds. So thank you for your attention, and maybe there's time for questions.